nanohub.org.
Online simulation, and more for nanotechnology.
Today's topic will be wave particle interaction characterization, and specifically we will be talking about confocal microscopy. Our first speaker will be Dr. Fatma Pri-Cakmak. She's actually a postdoctoral researcher. She's at MIT in Boston, and you may recognize her last name. She and our Dr. Cakmak is actually our couple, a distance couple right now. Penn State, here in State College, Pennsylvania, and Fatma is in Boston, Massachusetts. So we're really excited to have Fatma here today.
So I'm going to talk about confocal microscope today and my research during my PhD, mostly at Penn State. But before going further into confocal microscopes, I want to talk about size of the things we are looking at, right? Why do we need in the first place a microscope? Because the things we want to look at are quite small. If you think a solar system we are living in, it's like 1 billion meters, such a huge system. If you just like think Boston and Providence, it's just like 100,000 kilometers, and still like a size that we can gather. And humans are in the range of like a couple of meters. And if you look at a cell size is around, according to cell, it changes around like 100 micrometer, and it's also like around 100,000 meters. So, but like we can see cells, but we also want to look the molecules inside the cells. They are even smaller. They are actually around 1/billion meters. They're around nanometer range. So what did people, humankind, come up as a solution? Microscopes. When you think about the microscope, actually as this little cute kid is holding, the lens is the key. Why do I say so? Because the first people discovered lenses, and that's how it started forming a microscope. If you think when the first microscope is founded, it's around 1595. And it is founded by Hans and Janssen. And this is the first compound microscope. It just has a simple lens adapted at the ends of the tube, and it can only enlarge objects 3 or 9 times more. You can see here the lenses, and this is just like a tube. That's how it started. This is, of course, still very exciting discovery. This is the start of how we are looking at these small things, right? And then when we think more complicated, since this was a start, but like people didn't stop there, because we want to look things in more magnification, and this required more sophisticated methods. And in almost like 100 years later, in 1665, Robert Hooke used the term for the first time, cell. And this is something like a microscope he used, a simple microscope still, but still advanced version when you compare to the first microscope I just showed you. And he used cells because he thought he saw cells. And when he was looking actually cork, he was actually looking at the bottles in the cork, and he thought about the rooms where the priest stayed, and he decided to use this name to describe what he was seeing. This is like what he actually observed under the microscope, these little rooms, and it reminded him the rooms where the priest stayed. And then actually, the first person who observed real cells is immediately after Hooke, Anton Leeuwenhoek, Dutch guy, amateur scientist, so people actually snubbed him. He actually had a different name for the cells, but they didn't use his definition. They went with the academic person who was the Hooke. So they described when they look at living cells, they use the cell as a definition. And this is actually very interesting because this is like the microscope Anton von used. This is such a small, tiny microscope, which is kind of very amazing, right? How far we have come. So this is just basic lenses and just like knobs, and this is like people are really good in engineering at the time, whatever they have, and they were able to like form these little small things, and so they were able to like observe not a great resolution or anything, but just like whatever they can get their hands off, because it was very laborious and they didn't have the technology we have today. So we came to the modern microscopes, right? We start from very simple thing. The engineers and physicists, they just build up all those things together, and then we came up with this modern microscope. Modern microscope actually has tons of lenses instead of like, if you remember, I just showed you in the first one, you had 2 lenses, very simple, and you were able to only just magnify things 3 or 9 times more. But when you have the modern microscope, we have this technology right now, so we are able to like use this technology and put tons of lenses everywhere and like build this more complicated machines compared to the like first ones, and we can actually get 100 type magnification, and we can even get better resolution than the limit of light. So what you have here is that you have usually this is just a general microscope. I just show like this is just body of the microscope. You put your sample here, and this is your lens. You can actually look your sample under this, and this is a light source. This is actually a simple one, and so you can actually have other types. If you want to look at fluorescence microscope, you have other sources like laser powers. Just you can see here the light. It's coming through the sample. This is the stage, and you have all these other parts actually in the microscope helping you to get a better resolution. You can look at your sample from your eyepiece. You have the objective here, and objective determines like your refractive index, your numerical aperture, and this also very important determining the resolution you will observe. So these are condenser, it just condenses the light, and you have filter diaphragm. It helps you to just choose a light how much you want. You can actually have different filters, and you can have a collector lens, and you can actually just use a lamp house, or actually you can just use at these days they use just lasers to as a light source. And you will have you can actually have a computer just to see, just reflect whatever you are seeing. You can actually focus your sample. You can have all these things, and it's actually easy to customize all these things for your sample. As I said, you are actually have this like all these eyepiece, filter cubes, and you can have the lasers, and you have your sample here, and you have the computer, and whatever you see here, and you can see here. So just in case like everybody is not familiar with the fluorescence, fluorescence is just when you excite a sample, dye molecule or whatever you have in your sample, and it will emit light, and you can collect this light by using this technology. By using the same PET light, you're exciting the sample. So you have this camera, and you can actually just collect all of this data. You have these detectors, and you can collect all this data, and you can just turn to an image, and this image will give you details about your fluorescence intensity in your sample. And whatever you label, for instance, you are labeling a biomolecule in a like cell, and then you will see the biomolecule you labeled in the cell where exactly it's located, and you can even see the concentration, because the labeling efficiency is gonna whatever you labeled is, if it is like higher light intensity is there, it indicates you have more dye molecules there. If it's a lower fluorescence efficiency, fluorescence intensity, you will see like less like a dim signal, and you can actually get quite quantitative data out of this. So this is just an example like you see here, but you can use different labels and just look at different properties of the molecules. So what is the special thing about laser scanning confocal microscopy, if you ask? Actually, now the microscopes all have very like standard things. But for the scanning confocal microscope, I'm not talking about very fancy ones, but just like a just general in terms of if someone asks you the thing you should know about the laser scanning confocal microscopy is that you need a pinhole. Pinhole is the reason laser scanning confocal microscopy has a better resolution. People prefer to use this one. What does it mean, right? So if you think about like you have your sample here, right? And you're just exciting the sample with a laser light, right? And then you're collecting this very dim signal coming out of your sample. And all this dim signal is coming at the same time. If you look at all your sample, if you look at just epifluorescence, just a regular fluorescence microscope, it will come all at the same time. And this is what's going to happen is that you cannot differentiate between the slices of layers of the sample you have. Because think about a cell. Cell has a wide, like it's not just one slice, right? You have all these heterogeneous systems just being distributed all over the cell. Like think about the thickness of the cell, right? You will have different properties, all this thickness. If you want to just look at the one slice of the cell or the sample, you will need to focus on that part. And you want to just collect the data coming out of that. How can you do this? And ingenuity of people, mankind, it shows up. You basically put this like pinhole aperture, right? You still get signals from the sample at the same time. But with this pinhole, actually, you're only collecting the certain plane you want to focus on. So for instance, you have all these planes, let's say 3 planes that are coming all of them, but you can actually make this whole, because the signals coming from 2 other planes is not going to be able to come back to the detector. So you're only going to see the signals coming only from one plane. You can see here like very nice illustration, right? See, it is just coming on the one plane. Other 2 planes are just like gonna be not collected from the detector. It's still there. You're not doing anything to your sample. It's still there, but you're just collecting some certain region, and it actually lets you just slice up like your sample. It's quite nice. You're just slicing up and you're just observing that part of your sample. I think quite simple method. You just have a hole. You just put this like little quiet hole, and you can actually adjust the hole if you want to have like a smaller like thickness. You can have a smaller pinhole, and then if you want a bigger one, you can actually open. I had to actually do experiment in one time. I had to open this hole all the way because I wanted to collect the signals from all of the sample. This is just further showing that illustration how wide field versus point scanning differs. As I said, when you have the wide field scanning, you have all the signals coming from everywhere. But when you have the point scanning, you will have this just tiny region you are scanning, and it will help you just localize the signal better. And this actually helps you a lot, right? Because you are able to look at like a certain slice, and it gives you better resolution, and how this like reflects in the images. So if you look at a wide field epifluorescence wide field microscopy, you will see the signals, all of them coming together at the same time. Like you will see all the samples, right? For instance, just like focus on this red one. It's like maybe a virus particle or something like that. You are just seeing the outside of this structure. But if you use the confocal microscopy, you will able to slice. For instance, you will able to see the slice like this here. You will able to also collect the data inside of the sample as well, and you will have a better resolution because you're actually looking at like a tinier plane, and it will help you to get more information about your sample. That's why people actually want to use confocal microscopy. So I mentioned about like the background, how people use the confocal microscopy. So, and you can actually do interesting experiments with the confocal microscopy, such as diffusion experiments. And I'm going to go a little in details. what I have done for my PhD at Penn State. I will talk about diffusion of biomolecules, how you can calculate this in a research setting. Just to give a few background, I'm going to talk about compartments. If you look at the cell, right, cell itself is a compartment. It also has further compartments by membrane-bound or membraneless organelles. And these membraneless organelles reported to have liquid-like characteristic. And it's a recent discovery. People are really, really interested in this compartments. For instance, activities in cell can be compartmentalized further by phase separation. And people reported number of cytoplasmic and nucleoplasmic membraneless organelles composed of RNA and protein. And this is quite exciting because this can help you have biomolecular partitioning, co-localization of the, like for instance, you can have something different in this compartment and you can have a different molecule in the other compartment. And this can also help with regulation of metabolism and so many different biological concepts. So it's kind of important to have this like little compartments. And what I was doing was I was creating these compartments in vitro. In vivo, people are actually observing this phenomenon, and it's like very interesting, and it has been indicated in many disease and cellular functions. So I was able to recreate this in vitro, meaning that in test tube, not in the cell, but in the test tube. As I said, this phenomenon has been indicated in diseases such as like Alzheimer. You actually have this fibrillization formation in Alzheimer, and actually when you look at these things, they first have these liquid-like properties, and then they form these pathological aggregates. And it is interesting to understand how this transition from liquid-like structures to aggregates happens. That's what I was studying. So it's important to study the diffusion of biomolecules, because when you have liquid-like dynamic structure, you will have a faster recovery, but if you have an aggregate, you will have a slower recovery. And you can use to determine what kind of structures you have at time points by just studying their fluorescence recovery, just dynamic properties. This is the system I have used. I like to show structures. It might become a little scary, but it's not. I know engineers doesn't like chemical structures, but as a chemist, I love structures, and that actually makes sense for me. But basically, you don't have to think about the structures. Basically, we have these two different biomolecule, RNA and polyamine spermine, a positively charged molecule. And when I mix them, I can actually form this mimicking compartments we observe in the cell in the in vitro systems. So when I mix them, they look like this turbid solution. If I centrifuge them, they are actually very, very small, tiny droplets. And this is like how they look like under the microscope. That you have these droplets you see here. Let's say condensate droplet, because coacervate is kind of difficult to pronounce it. I think condensate is going to help people to just say it even easier. So I use these biomolecules just mimic the compartments in vivo system, and this is how they look under the microscope. So when you have these condensates, you can actually put more biomolecules, and they will selectively go to the droplet itself, because of the charge interactions, also other interactions involved, right? So you have the, let's say, like red fluorescent labeled another RNA piece, and see, you just add this solution, it will selectively go into the droplet. And this is what it looks like. If you have like a coacervate droplet, you have a bright field image here, just meaning you have a bright field, nothing in fluorescence, you're just looking at like through light, and you're actually looking here in the fluorescence signal, which means that I'm using, for I think it was 647 certain excitation wavelength, and then I'm looking at the emission coming out of my droplets, and this is how they look like. You can see that whatever I used a labeled RNA, and it's partitioned into the droplet. It's a perfect match with the droplets you see in the bright field and the fluorescence. And you can actually calculate the concentration of these droplets in the concentration of the biomolecule you added in the droplets by just using calibration curves. Basically, you just make a calibration curve to obtain the concentration value. You made like this graph and like with known concentration. By using this graph, you calculate the concentration of the biomolecule partitioned into the system. I actually use 3 different biomolecules. First one is polyuridine. It is 15 mer, 15 repeats of nucleotides composed of uridine. And the second one is poly-M15, again 15 nucleotides and composed of random nucleotides. And I also use a poly-A15, again 15, same size, same length, but this time it's composed of adenosine. As you remember, I was using PolyU and some spermine, like positively charged molecule. PolyU is also the same thing, exact like and it's the U. So when I look at them, it has the weakest binding with PolyU, right? Because it's exactly the same thing. When you partition this like labeled molecules, these are the strongest binding because, as if you remember from your biology class, it has it's a base pairing. A and T and G and C. They have this like base pairing through hydrogen bonding, so they have the strongest binding. When I calculate the concentration in the droplet and the concentration in the supernatant, I can actually calculate how much my biomolecule is concentrated in the droplet. It is a lot higher when I use A15, which is expected because it has the strongest binding. 2,800 times more. When I have the PolyU, it actually is around 44 times more partitioned into droplet. It's a quite stark difference. It shows that like you have certain interactions to have these things to partition, such as base pairing. So I'm going to talk about FRAP. What's FRAP? FRAP is the fluorescence recovery after photobleach. So I'm going to talk about 2 different types of experiments I have done. First one is partial bleaching. I just bleached the certain part of the droplet. So when I'm saying bleaching is meaning that I use very high power of the laser, just get rid of all, like bleach, erase the fluorescence in that region. And it looks like this. Afterwards, you choose a droplet of interest and you just bleach that region you want to just a part of the droplet, and then you look at the monitor the recovery, and this gives you information about the diffusion of the biomolecules, because when you bleach this is actually not coming back. How you get a recovery in this region is that, if you can see my cursor, it's coming from outside. Around the like other labels are coming in. So this lets you know how fast these biomolecules are coming in, and you can actually get the diffusion by using this information. And I use those entire droplet bleaching. You have this droplet, you just bleach all of the droplet. You use like a higher region of interest. You can choose the region of interest by like using this microscope software. And then you look at the, again, recovery, and this time it's actually between the droplets, how they communicate with each other and how the other biomolecules is coming in. Because when you bleach, again, it's not gonna come in within the droplet. It's not recovering from the bleached molecule.
But just a quick question. Are you changing the pinholes while you're doing this partial bleaching to full bleaching?
I usually don't change within the same experiment. It's a good question, but I usually don't change within the same experiment, just to keep the parameters same so I can compare within the sample. You choose it just for the like for instance for this droplet type, I choose a certain pinhole, and usually people use like one atomic unit, like the size of the pinhole, and it's kind of useful. And it also determines the resolution too. Like you don't want to go like too high, and you just want to keep similar pinhole size for your experiments. So for the FRAP, actually for the FRAP graph, you have this is just the general FRAP graph I have here. don't be scared of equations. They are simple equations. I'm going to talk a little about it. So as I said, you have like a droplet of interest, and you photobleach this droplet, and you look at the monitor, you monitor the fluorescence recovery, right? So you have this fluorescence before bleaching, and it goes to like, it doesn't have to go zero, like just like say it goes to zero, and you look at the recovery, and this is the curve you will get. This is just recording the fluorescence for the region of interest. This is quite like a nice graph, right? And now by fitting this graph, you can get half time of recovery, which is the time where fluorescence is recovered to half of its maximum value here. And you can use this information to calculate, you get half time, you use this information, and you can get this diffusion coefficient by using this half time. And this diffusion coefficient lets you know how fast your things actually diffusing in and out. So when I actually look at these samples, what I observe for this 3 different type of biomolecules I have looked, they actually, if I do the partial droplet recovery, it's quite fast. It doesn't really matter what type you have. It diffuses because it's within the molecule, within the droplet, it diffuses so fast and it recovers quite fast. And if you look at the diffusion coefficient, as you see, it's like very similar. Again, you actually even fastest because I think the interactions are not strong enough. It's not attached, and it's like more free to move around. Let's look at the entire droplet recovery. It's completely different story, right? You have three different molecules, A15, M15, and U15. If you look at the recovery, I'm talking about 1,000 seconds to 10 seconds compared to a U15. This takes A15 being, as we know, right, it's interacting strongly, so recovery takes long time. But if you look at the U15, again, the interactions are not as strong, and the recovery takes a couple of seconds. And M15 is between, as we expected. And this is something we observe for their diffusion coefficient, right? The diffusion coefficient for A15 is quite small, and U15 has, again, almost same or similar diffusion coefficient for entire droplet to the partial droplet. But we can add actually vesicles. Vesicles are just like lipid molecules, just and around this coacervate droplet, and see whether they form a bilayer or not. By just seeing how their dynamic properties changed. So you have these droplets, you have the vesicles, and when you look at these vesicles before, I'm photobleaching just this region here, shown in red, shown in green, and if you look at them, they don't recover at all. It shows that these vesicles I have put around my droplet actually are not connected, and there's no diffusion between this between these vesicles themselves. Just to show you, there is also doesn't necessarily have to be recovery, right? I show you how it recovers, but it doesn't necessarily have to recover. So I also look when I have this these vesicles around my droplet, whether this impede the diffusion, and I just photobleach all of the droplet for these samples, and RNA diffusion into and out of the causal droplets wasn't impeded. You can actually see here, I'm using U15 in this case. You can see the very rapid recovery. Again, very similar. So with that, I actually show you that I can form compartment mimics to study cell compartmentalization in vitro. And I show that rapid exchange of oligonucleotides observed for partial droplet bleaching, but for entire droplet recovery dependent on binding interactions and supernatant phase concentration. And just accumulating lipid vesicles around the coacervates did not result in inhibit of diffusion of biomolecules. I just show most of my stuff from Penn State, so I would like to thank my group, my advisor Chris Keating, and all this very funding agencies, NSF and NASA. I was mostly funded by NASA, so it's quite cool to just study these things, because this is also mimics for how cells might have evolved from the first cell. Also, this mimics can also use for protocell models. With that, I will be happy to answer if you have any questions. Thank you.
Fatma, this is Peter. I've got a question about just doing the base microscopy on the droplets. Do you need to use a fixative in order to prevent the droplets from moving around, or do the droplets on their own just stay in place when you do the imaging?
They actually do stay in the place. Yeah, they are quite big, like 10 micron, 20 micron. So they're not affected by the Brownian motion, so they are quite big. So, and I'm also using inverted microscope, so they are actually settled down at the bottom. I'm looking at from the bottom too. That also helps.
Wild question, Fatma. Maybe it's very irrelevant, but would we be theoretically able to also get diffusion constant for the COVID penetrating into the cell membrane in a similar way? What do you think?
Can track it, but I wouldn't say it's like a diffusion coefficient, because it's already like it's a protein. I mean, and it's interacting with many things at the same time, and it's actually reforming this other proteins, or also just like forming these endosomal vesicles inside, and you're just going to the like cell nucleus and like make the cell nucleus, depends on like what kind of virus you have. It's gonna make cell nucleus work for the virus and make more viruses, and it will bother and like come off. It's gonna be probably a longer process, I would say, and usually I'm actually I'm going to work with viruses, but basically what people do is just track them. It forms quite interesting phenomena in the cell, actually. It does like make endoplasmic reticulum to restructure and just use all these cellular mechanisms for its own sake. So, yeah.